DNA is the ultimate directive for cells and it codes for your traits. In preparation for cell division, DNA replication occurs so that each of the new daughter cells have DNA. In eukaryotic cells, DNA replication occurs in a nucleus, while prokaryotic cells, which don't have a nucleus, replicate DNA differently. DNA, or deoxyribonucleic acid, is composed of a deoxyribose molecule bound to a phosphate group and various nitrogenous bases: thymine, guanine, adenine, and cytosine. Adenine and guanine are purines, which are two ring structures, and thymine and cytosine are pyrimidines, which is one ring.
Complementary base pairing ensures that the two daughter molecules are exact copies of the parental molecule. When the parental molecules separate, each serves as a template on which nucleotides are arranged into new complementary strands. The replication of chromosomal DNA begins at the origin of replication, which has a specific sequence of nucleotides. Many enzymes participate in the process of replicating the DNA strands. The proteins that initiate DNA replication recognize the sequence in order to begin separating the two strands to make a replication bubble. At the end of the bubble is a replication fork, a Y-shaped region where the strands of DNA are being undone, almost like a zipper.
DNA helicase, an enzyme, has an essential role during DNA replication for the separation of the double-stranded DNA into the leading and lagging strands as templates to be copied. The DNA helicase unwinds the DNA at the replication fork, using ATP to break the nucleotide bonds. The unwinding of the strands causes them to twist tighter and strain the replication fork. The topoimerase relieves the strain by breaking and rejoining the DNA strands. At the separated strands, primase synthesizes RNA primers using the parental DNA strand as a template.
The lagging strand is synthesized discontinuously in a series of fragments, also known as Okazaki fragments. The strands are anti-parallel, meaning they run in different directions of replication, so the lagging strand runs in a 3' to 5' direction, while the leading strand replicates continuously in a 5' to 3' direction. During repair synthesis, a DNA polymerase replaces missing nucleotides, using an undamaged strand as a template. To complete the new strand, DNA ligase seals the ends of the new DNA to the old DNA. DNA ligase will appear specifically in the S phase of replication, while ligase repairs any damage to the DNA at any stage of DNA replication.